Hi students, this is A. Mandari Kumar Rajesh. In this video, we are going to discuss the topic called gram staining procedure. Before we get into the topic, I would like to give you a brief introduction about the different types of staining process with a flow chart. So, staining process can be broadly classified into two types. They are simple staining process, then differential staining process. In simple staining process, a single reagent is used. A single staining reagent is used to stain the cell structure with a simple staining procedure. The simple same staining procedure, and that process is called as simple staining. While in the case of differential staining process, more than one reagent staining is used to stain the cell structure, and it exhibit different staining procedure, and that process is called as differential staining process. Then simple staining process again classified into two types. They are positive staining procedure, then negative staining procedure or negative staining process. In positive staining process, the stains are positively charged. And the stains can attach to the surface of negatively charged object. So the stains are positively charged. It can attach to the surface of negatively charged object, and that process is called as positive staining. While in the case of negative staining, the stain contains negatively charged and it repels from the negatively charged objects, and the stain can fill the space between the object. So in negative staining, the stain contains negatively charged and they repel from the negatively charged object, and it fills the space between the object, and that is called negative staining. Then differential staining process again classified into two types. They are separation of microbes into different classes. Separation of microbes into different groups. Then the second one is visualization of various structures. Then again, the separation of microbes into different groups again classified into two types. They are gram staining procedure, then acid fast staining procedure. So in this video, we are going to mainly concentrate on gram staining procedure. The acid fast staining procedures are used to identify tuberculosis bacteria, that is Mycobacterium tuberculosis. So acid fast staining procedures are used to identify tuberculosis bacteria. Then visualization of various structure again classified into five types. They are wall staining procedure, spore staining procedure, then capsule staining procedure, then nucleus staining procedure, then finally the flagellar staining procedure. So the visualization of various structure can be classified into wall staining procedure, spore staining procedure, capsule staining procedure, nucleus staining procedure, then flagellar staining procedure. So in this video we are going to discuss the gram staining procedure. So the gram staining procedure was developed by a Danish physician named Christian Gram, namely Christian Gram, in the year 1884. So the gram staining procedure was developed by a Danish physician Christian Gram in 1884. By using gram staining procedure, we can classify bacteria into two classes. They are gram positive bacteria, then gram negative bacteria. And this gram staining procedure comes under differential staining procedure. So the gram staining procedure which comes under differential staining procedure, that means in gram staining procedure we are going to use more than one reagent and it exhibits different staining procedure. So now I am going to tell about the steps which are involved in gram staining procedure. So please wait a minute. Let me explain the steps which are involved in gram staining procedure. The first step, we have to prepare a smear of bacterial culture. That means we have to prepare a streak of bacterial culture on a slide. Then stain with crystal violet for 30 seconds. We have to apply the stain on that slide for 30 seconds. Then rinse in distilled water for 2 seconds. Then gram iodine for 1 minute. After that we have to add gram iodine for 1 minute. So this is a differential staining process. So we have to use more than one reagent. So first we have used crystal violet. Now we have used gram iodine. Then rinse in the distilled water. Then wash that slide in 95% ethanol or acetone for 10 to 30 seconds. 30 seconds. If it is gram positive bacteria, it retains the crystal violet. It retains the crystal violet color. If it is a gram positive bacteria, after the treatment of the ethanol, 95% of ethanol or acetone, the bacteria cell wall retains the crystal violet. Then rinse in the distilled water. Then saffron for 30 to 60 seconds. Saffron is a stain, so we have to apply on the slide saffron for 30 to 60 seconds. Then rinse in distilled water and plot and observe under microscope. If you observe a slide under the microscope, it shows the crystal violet. That bacterial culture shows the crystal violet, and that bacteria is said to be gram positive bacteria. Suppose this, after the rinse in distilled water, that slide has to washed in 95% of ethanol or acetone. If it is gram negative bacteria, this ethanol removes the crystal violet stain, from the cell wall. Then rinse in distilled water. Then saffron are added to that slide. Now rinse in distilled water and plot, and now you can observe the slide, observe under microscope, and you can find the slide which shows red color, that means pink color. That means the bacteria is a gram negative bacteria, because after the treatment of alcohol, the crystal violet stains are removed from the cell wall of gram negative bacteria. So by this procedure we can differentiate gram positive and gram negative bacteria. Positive bacteria contains in the cell wall like granulars, that is granules. Then it contains teichoic acid and teichouronic acids. So by this composition of cell wall, they can retain the crystal violet. It can retain the crystal violet, and the gram positive bacteria cell wall is made up of peptidoglycan. So due to the chemical composition of the cell wall, it can retain the crystal violet color. While in the case of gram negative bacteria, the cell wall is poorly made up of the peptidoglycan. It contains small amount of peptidoglycan and it contains lipoprotein, then outer membrane and polysaccharides, more amount of polysaccharides. So due to the different chemical composition of the cell wall, the gram negative bacteria are not able to retain the crystal violet. They are not able to retain the crystal violet. Now I am going to tell about the difference between the gram positive and gram negative bacteria. Let me explain the difference between gram positive bacteria and gram negative bacteria. So here I have written the differences between gram positive bacteria and gram negative bacteria. So first characteristic feature, cell wall. In gram positive bacteria, single layered with 0.015 mu micron to 0.02 mu micron. So thick, 0.02 mu micron thick. While in the case of gram negative bacteria, they are triple layered. That means outer membrane, lipoprotein and lipopolysaccharide. So they are triple layered with 0.0075 mu micron to 0.012 mu micron thick. So the gram positive bacteria cell wall is somewhat thicker than gram negative bacteria. Then rigidity of the cell wall. So gram positive bacteria shows the presence of peptidoglycan. So the cell wall is rigid. So rigid due to the presence of peptidoglycan. Here elastic due to presence of lipoprotein and polysaccharide mixture. So in the gram negative bacteria, the cell wall is elastic due to the presence of lipoprotein and polysaccharide mixture. Then chemical composition of the cell wall. So the cell wall is made up of peptidoglycan and it contains 80%, polysaccharide 20% and teichoic acids are present. In the cell wall of gram positive bacteria, peptidoglycan, the cell wall contains peptidoglycan 80%, polysaccharide 20% and teichoic acids are present. While in the case of gram negative bacteria, the cell wall contains peptidoglycan 3 to 12% only, rest in polysaccharides and lipoproteins. So in gram negative bacteria, the cell wall contains smaller amount of peptidoglycan and teichoic acids are absent. Teichoic acid is absent. Then outer membrane is absent in gram positive bacteria, while in the case of gram negative bacteria, outer membrane is present. Then periplasmic space. Periplasmic space is nothing but the space between inner cytoplasmic membrane and outer cytoplasmic membrane, and that space is called as periplasmic space. So periplasmic space are filled with matrices. Then these periplasmic spaces are present in gram negative bacteria. The periplasmic spaces present in gram negative bacteria. In the case of gram positive bacteria, it contains periplasmic space much smaller than gram negative bacteria, but in our book it given absent, but the gram positive bacteria contains periplasmic space, but they are much smaller than gram negative bacteria periplasmic space. Then susceptibility to penicillin. So the gram positive bacteria are susceptible to penicillin. So penicillin is an antibiotic. It can kill the gram-positive bacteria due to the rigid cell wall. While in the case of gram-negative bacteria, they are low susceptible because they are elastic in nature. The cell wall is elastic in nature, and due to the different composition of cell wall, they are low susceptible to antibiotic penicillin. Then nutritional requirements. In the case of gram-positive bacteria, it is relatively complex. Due to this type of nutrition, we can easily kill the gram-positive bacteria. While in the case of gram-negative bacteria, relatively simple. They need nutrition very simple. They can absorb nutrition very by using simple processes. While in the case of gram-positive bacteria, it can absorb the nutrition by complex processes. So we are not able to kill the gram-negative bacteria very easily. Then flagella. In gram-positive bacteria, it contains 2 basal body rings, and in the case of gram-negative bacteria, it contains 4 basal body rings. Then lipid and lipoproteins are low. They are very low in gram-positive bacteria. Gram-negative bacteria contains high amount of lipid and lipoproteins. Then lipopolysaccharides are absent in gram-positive bacteria. Gram-negative bacteria contains the lipopolysaccharide. So these are the difference between gram-positive bacteria and gram-negative bacteria. So that's all about today's video. If you have any doubt, please clarify with me. Thank you very much for watching this video. Thank you.